Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are working our way through Chapter 5, and we are working our way through cellular respiration. Gave an introduction about that. We divided it into 4 stages. Did you write them down? Because we're going to refer back to it. The first stage is glycolysis. So that's what we're going to talk about in this video. All right, so glycolysis is literally glyco for sugar and lysis. So we're actually breaking sugar, splitting sugar. We're going to start with glucose. This is the very first step. We're starting with glucose, and we're going to start to split it apart. It's also got another name, Epstein-Meyer pathway. Actually, I think they've added a third name with that. These are named after the people who figured this out. But most of the time you'll hear it referred to as glycolysis. Glycolysis is the metabolic pathway that occurs first. It is first for cellular respiration. It's first for aerobic cellular respiration, and it's also the first stage in anaerobic cellular respiration. What's the difference between those two words? Oxygen. Aerobics requires oxygen. Anaerobic respiration does not use oxygen, but it's the exact same process. So as we go through respiration, I want you to be looking for where oxygen shows up. Because that's going to make a difference of aerobic to anaerobic respiration. So be watching out for oxygen as we go through this. Glycolysis is also the first step for a metabolic pathway called fermentation. We've heard about fermentation because of Pasteur and his grape juice and wine experiments, and we have found that yeast have the right enzymes that they can take the precursor, sugar, glucose, from grapes and produce an alcohol end product that gives you wine. So this is a fermentation pathway, and we'll talk about these two in this course. So let's talk about glycolysis. Glycolysis itself is anaerobic, so no oxygen in this part of this metabolic pathway. It is a pathway, so we're going to require some steps. Glucose is the precursor, and it has 10 enzymes. Your end product is going to end up being pyruvic acid, and we'll see how that works into it. Okay, so I'm going to switch to my whiteboard, and we'll see how this goes, and hopefully you can see it. The good news with videos is you can screenshot, so that'll help, but I usually write this part in class. So let's start with glycolysis. The precursor for glycolysis is glucose. So we're starting with glucose, and we broke that down already. If this was a human, you took it in, your stomach breaks it down, digestion gets into glucose, which enters your bloodstream, goes to your cells, which is where glycolysis is going to occur. Bacteria just goes right in, glucose. And the first thing that's going to happen in glycolysis is we're actually going to add some ATP to glucose. Which is kind of strange because we're trying to make ATP, and the end product of cellular respiration is ATP. So it's kind of sad that we're using up some at the beginning. But have you heard that phrase, you need to spend money to make money? You need to invest in your business. That's what's happening right now. We're going to take 2 ATP and invest into this. We're going to take ATP. We're going to take the phosphates. The phosphates ended up getting added to glucose, and then it becomes adenosine diphosphate, which can later on become adenosine triphosphate again. So the first thing we need to know is that in glycolysis, we have to put into ATP, which is going to also be important because just like business, and when you invest money in order to figure out your profits at the end, in order to figure out how much ATP we made at the end, we need to remember that we invested some at the beginning. So keep that in mind. All right, so we're going to put this in. The phosphate groups actually get added to glucose. It becomes one of those long names, so I'm not going to make you remember. But know that this is an investment stage. This is called substrate level phosphorylation. We're going to phosphorylate this substrate. So those are some things that happen. What else do you need to know? Okay, so now we're going to get busy making ATP. So some other things occur at this, and one of the things that is going to occur is that we're going to have some oxidation and reduction reactions. Remember sharing those electrons, picking up electrons. NAD plus, nicotinamide adenine diphosphate, is a factor that's really good at picking it up. It's one of those vitamins that we have to take in from that helps make this work. Remember, we referred to that. NAD plus is capable of picking up some electrons from glucose. So glucose, so NAD plus is going to pick up some hydrogens and become NADH. Where did those hydrogens come from? Well, they came from glucose. Now, just remember glucose, I drew 6 circles up here. Those represent my 6 carbons, just kind of a shorthand. There's my 6 carbons for glucose. But remember, glucose had 12 hydrogens, right? 6 carbons, C6H12. So a couple of those hydrogens are going to get picked up here. And we're actually going to do that twice. 2 NAD pluses are going to pick it up. So we're going to get 2 NADHs occur from that. This is called, remember we're sharing electrons, NAD plus picked up, accepted an electron with its hydrogen, oil rig. Oxidation involves loss, reduction involves gain. NAD plus got oxidized or reduced. I'll let you guess. NAD plus got reduced. Reduction involves gain and you can see it. But if something got reduced, something got oxidized. Who donated the electrons? The carbon molecule. Now it's not called glucose anymore, it's got another name for it, so we're just going to refer to it as the carbon molecule, but the carbon molecule got oxidized. It donated these hydrogens. So we just had some oxidation and reduction reactions happen. That happens in glycolysis, and it's going to happen twice in glycolysis. So you need to know that. So we've got some oxidation and reduction. The glucose molecule, the carbon molecule, is getting oxidized. What else is going to happen? We actually make some ATP during this process, and we actually make, oh, I didn't draw that right. We actually make 4 ATP in this process. So I also, my arrow got wonky, but we're also going to make 4 ATP. So that's exciting. We make 4 ATP during this process. So that's good because we're trying to make ATP. So we do make a little bit of ATP. And at the end of glycolysis, we started with 6 carbons. We still have all of our carbons, but they have been broken up into 3 carbon molecules called pyruvic acid. And we get 2 of them from this. So our 6 carbon molecule got broken down, catabolized during this process into 2 3 carbon molecules called pyruvic acid. So pyruvic acid is your end product from glycolysis. Starts precursor is glucose, your end product is pyruvic acid. Now, you may have learned this before. You may have learned the word pyruvate. Pyruvate and pyruvic acid are one of those molecules that can go back and forth between an acidic version and a neutral version. So the neutral version is called pyruvate. The acidic version is called pyruvic acid. Your book that we're using uses pyruvic acid, so that's what I'm going to use. But no, it's the same. It's not the same thing. It just rotates back and forth between that pyruvate and pyruvic acid. So you may have learned this as pyruvate, or you may learn it in the future as pyruvate. So that's what's going on there. Okay, so we just finished glycolysis. 6 carbon glucose precursor. We invested 2 molecules of ATP. We phosphorylated glucose into an intermediate. We had some other intermediates. During this time, we had oxidation and reduction. NAD plus came up and became NADH. We actually made 4 ATP. Now, if you're keeping track, we do need to subtract the 2 ATP. So if I ask how many molecules of ATP are made during glycolysis, well, we made 4. If I ask what is the net number, which means to get net, you have to take 4 and subtract your initial investment. What's your profit from that? What is the net result? How many ATP at the end of this did you end up producing? Well, you ended up producing 2 because you had to use 2 to begin with. You had to subtract that. So keep that in mind. Hey, so I've got a molecule that has some hydrogens. Can you think of a place where we need a lot of hydrogens during cellular respiration? I'm hoping you remember chemiosmosis and all those hydrogens that are needed. So this NADH is going to take his hydrogens to the electron transport chain. He's picked some up. We need some hydrogens for the process of chemiosmosis, and that is going to occur at the electron transport chain. So we're starting to see where this is all going. Okay, so let's go back to our PowerPoint and see what we need to catch up on. Input, I'm going to give you a second and see if you can fill this in. Input, 2 ATP inputted in. We have Oxidation and reduction happens. The carbon molecule, the food, got oxidized. NAD plus got reduced to form NADH, and that happened twice. We made 4 ATP, but there was only 2 net because we had to subtract what we started with. The end product is 2 pyruvic acids, and you can see we have C3H4O3 times 2, so I still have my 6 carbons. I still have my 6 oxygens. What I've lost is some hydrogens, and those were picked up by, these oxidation reduction reactions occurring with NAD+. Remember, glycolysis is the first step for aerobic respiration, anaerobic respiration, and fermentation. Okay, keep in mind there are proper names for all of these intermediates. I'm not going to make you learn all these proper names. It is a metabolic pathway. You should know the precursor. You should know the end product. You should know what is happening during this process. 2 ATPs in, you can see how that happens, but I'm not going to make you memorize the proper names for this. Someone, if you've got big goals and you've got biochemistry, organic chemistry in your future, somebody's going to make you memorize this. But for right now and for what you probably need, this is more important to understand how it works. So don't be scared of proper names, but you can see how it relates. Okay, fill in your notes. Take a stab at it. How'd you do? Glucose is the precursor for glycolysis. Glucose is oxidized. It's really the carbon molecule as we're going through. It gets oxidized. The end products are pyruvic acid. Now, we have broken our $100 bill, but we still have like a 50. You know, McDonald's is still not going to take it. The vending machine's still not going to take it. Our cells still can't use it. We have a lot of our energy still locked up in our pyruvic acid. We did make a little bit. We made some ATP. So you got $1 you can spend at the vending machine. You got a little bit of ATP. Actually, I guess you have $2, right? Because you netted 2 ATP molecules. But a lot of the energy is still left in these pyruvic acids. Pyruvic acids are going to now become the precursor for our next metabolic pathway, our next stage, depending on whether you're going down the respiration or they can also be used in fermentation. We'll see how that works together. Okay, so we will stop that there and then pick up with what happens to pyruvic acid in the next video.